Hello boys, welcome back to another interesting session of video tutorials. This session is meant for class 12th biology students and this is for 14th September 2020, and we have been discussing about transgenic plants and also the biological applications, biotechnological applications in medicine. And this session we are going to discuss about another interesting topic, transgenic animals. We were talking about how to improve the plants and their products, how to make them plants pest resistant, silencing of mRNA we studied. Similarly, golden rice, incorporating vitamin A into the rice so that the nutritional malnutrition will not be there. Naturally, when we consume the foods that we get from the plant products, naturally we get all the minerals and vitamins. And for that, we have discussed how to incorporate a gene. So similarly, in animal products, we wanted to obtain animal products that help us to have good nutrition among us, plus we can increase their numbers. Whenever there is wildlife animals, particularly their numbers are coming down, they become endangered. So in order to stop endangered animals into extinct animals, we can use this biotechnology. So all these things help the animals to increase their numbers and they live healthily. And so we adapt biotechnology in this. So transgenic animals can be defined as animals that have their DNA manipulated to possess and express a foreign gene are known as transgenic animals. Of course, the moment you have seen this photo, you remember that rosy first cow, okay? And of course, you can see pigs, mice, fish, salmon particularly. All these are products of biotechnology. And what is that? Why should we do this biotechnology in transgenic animals? Reasons, many reasons are there. First, normal physiology and development. Transgenic animals are designed to study different aspects of genes, like how genes are regulated, how they affect normal functioning of the body and its development. For example, study of insulin-like growth factor involved in growth can be done by introducing genes from other species that alter the formation of this factor and study the effect of that results. Also, information is obtained about biological role of that steady insulin-like growth factor in our body. Study of diseases. Transgenic animals, which are models for human diseases, in which investigations of new treatments for diseases also gives understanding how genes contribute to development of diseases. So this is a major factor. Many genetic diseases, I have been telling you, many genetic diseases can be cured with this. So transgenic animals can become models in curing diseases such as cancer, cystic fibrosis, where the muscle fibers will be coming off. They will not be together. The muscle fibers, myofibrils will not be together like a bundle. Instead, as one puts on age, they come off. So that's cystic fibrosis. And rheumatoid arthritis, very severe condition where joints will have severe pain with a lot of fever. And of course, Alzheimer's disease. And all these which are incurable so far, now thanks to biotechnology, we are able to cure them. And what are the third aspect? The biological products. Products produced by living organisms, some of which are medicines, are very expensive. Even for now COVID-19 treatment, most of you must have heard that whenever a person with COVID positive is admitting into the hospital, particularly private hospitals, the cost of that would be around 4 to 5 lakhs, which will be very, very expensive for a normal person, for an average income person. So with the invention of this biotechnology, I think we are able to cure such diseases with less money. So that is our aim. Transgenic animals can be created by the introduction of genes that codes for particular product. like induces organisms to produce desirable product in the medium. Example, human protein to treat emphysema. Emphysema is a lung disease, already you know, accumulation of water in the lungs, emphysema. So there is a human protein that is alpha-1 antitrypsin. So that is the main aim or target for us. If we can introduce a gene into the human DNA which produces this antitrypsin, alpha-1 antitrypsin, I think naturally we will be having a cure for emphysema. Attempts are being made for the treatment of phenylketonuria. We were talking of this also, remember, in genetic disease, and cystic fibrosis. Sooner we will be having natural cure for all these incurable diseases. In 1997, the first transgenic cow, Roji, just now I said, first transgenic cow, Roji, produced human protein enriched milk. And how much? It is 2.4 grams per liter. It is protein rich. And the human alpha lactalbumin and nutritionally more balanced product for human babies than natural cow milk. So our effort is to increase the protein content, and this protein content 2.4 grams per liter, when we increase alpha lactalbumin, the protein, naturally the milk becomes richer by proteins, protein enriched milk. So compared to this, what normal cow produces, that milk will be lesser. Hence, thanks to biotechnology for introducing this in a cow called Roji, first transgenic cow. And fourth one, vaccine safety. For vaccine safety, vaccines are first tested on transgenic mice. Already again in COVID-19, you know, many companies, international companies, Bharat Biotech, they're all doing a lot of testing on COVID-19 vaccine. We are all eagerly waiting for that. And first they pick up the mice and they go closer to human. The latest we understand is monkeys. So the injection, the vaccine that is given, that COVID corona vaccine given to monkeys has produced a good result, and the monkeys are able to come out of the coronaviruses. So that's a good news for all of us. So in production of vaccines with safety, biotechnology plays a crucial role. For vaccine safety, vaccines are first tested on transgenic mice, developed for use in testing before they are used on humans. That's why we are getting delayed. Otherwise, see, somewhere 1 year back we had this corona and lakhs of people are getting dead, but still we are not taking any chances on human beings because it is to be tested and retested. Then only, once the results are fine, then only human trials will start. Transgenic mice are being used to test the safety of polio vaccine. And of course, just now I said, this is also for corona, COVID-19 vaccine also already tested. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. So ultimately, before it is given, any vaccine, before it is given to human beings, it will be last but one is monkeys, because we are all primates. And chemical safety testing. Transgenic animals introduced with genes which make them more sensitive than non-transgenic. They are then exposed to toxic substances to study the effect. Toxicity testing in such animals will allow us to obtain results in less time. So this is how we are able to do the various applications of biotechnology. So what we understand now is a transgenic animal contains in its genome a gene or genes introduced by one or other technique of transfection. The gene introduced by transfection is known as transgene. In animals, transfection specifies the introduction of a DNA segment, either naked or integrated into a vector into an animal cell. The same phenomena is known as transformation, as we have been talking about transformation. You can see various transgenic animals here. So the transgenic animals can be specifically designed to allow the study of how genes are regulated how they affect the normal functions of the body and its development. Study of complex factors involved in growth such as insulin-like growth factor. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of a disease. Transgenic animals that produce useful biological products can be created by the introduction of a portion of DNA or genes which codes for a particular product, such as just now I said human protein alpha-1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria, cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk. How much? 2.4 grams per liter. The milk contained the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural ones, natural cow milk. And transgenic mice, you can see here, transgenic mice are being developed for use in testing the safety of vaccine before they are used on human beings. Transgenic animals are made to carry genes that make them more sensitive to toxic substances than non-transgenic animals. So toxicity testing in such animals will allow us to obtain results in less time. Gene transfers have been successful in various fishes. I think somewhere I just showed you that, such as common carp, rainbow trout, Atlantic salmon. Let me show you that Atlantic salmon here. Yes, here you can see Atlantic salmon, and the catfish, goldfish, and zebrafish, etc. Genetically modified salmon was the first transgenic animal obtained for food production. Rabbits are quite promising. Rabbits are also quite promising in gene farming or molecular farming, which aims at the production of recoverable quantities of pharmaceutically or biologically important proteins encoded by the transgenes. Then goats. Goats are being evaluated as bioreactors. Some human genes have been introduced in goats and their expression achieved in mammary tissues. Transgenic sheep have been produced to achieve better growth and meat production. For example, human genes for blood clotting factor 9 and for alpha antitrypsin have been transferred in sheep and expressed in mammary tissues. Doggy is a transgenic pig, and it's a dog. Doggy is a dog, transgenic dog with excellent smelling power. See, dogs are known for smelling power, and when the biotechnology introduces the desired gene in the DNA of the dog has become better, so that it's directly helpful for police department. So doggy is a transgenic dog with excellent smelling power. So having known all this, we must be thankful to this baby of biology, namely biotechnology. Keep on listening